So proteins are an extremely diverse set of macromolecules. They account for more than 50% of the dry mass of most cells, and they have a very diverse set of functions in the body. You have your defensive proteins. They protect organisms from attack against disease. Organisms develop specific proteins to fight against specific diseases, such as antibodies. So antibodies play a big role in helping to keep us healthy, and they are a protein. Our enzymatic proteins increase the rates of our chemical reactions, and without these proteins, metabolism would grind to a halt, so our metabolic processes would not be able to occur. So enzymatic proteins are essential to life. Storage proteins. Storage proteins do things such as store amino acids, and albumin. Egg albumin is a common example of that. So eggs store the amino acids that they need in their albumin. Casein, the protein of milk, is the major source of amino acids for baby mammals. Plants have storage proteins in their seeds. Ova-albumin is the protein of the egg white, as we just discussed. We have hormonal proteins. Our hormonal proteins help in the coordination of all activities within our bodies. A prime example of a hormonal protein is insulin. And remember that amino acids and proteins are going to be hydrophilic. They are water loving, so they will dissolve in a liquid also. Unlike the steroidal proteins that we talked, or steroidal hormones that we talked about before when we talked about lipids. Contractile and motor proteins are essential to our movement. They allow our muscles to contract, and they allow the cells to move organelles around within the cell itself. So our contractile and motor proteins are critical for movement in both our life and within our cells. We have transport proteins. These are channel and carrier proteins that create ways for our hydrophilic and charged molecules to get across the cell membrane, because remember back when we talked with lipids, the cell membrane of a cell is made up of phospholipids, which have that hydrophobic area where the tails point in. And so anything that is charged or is a polar molecule or a large molecule needs a channel or a carrier in order to go into and out of the cell. And these are accomplished through our transport proteins. Receptor proteins, also a way for our water-soluble molecules and for the cell to detect stimuli. And so receptor proteins allow for responses to cells, chemicals in the cell, and to polar molecules to send a signal through that plasma membrane into the cell to help with cellular function. And then our structural proteins. These are what allow for the cells to be strong. Examples of structural proteins would be keratin in hair, silk and spider webs, collagen and elastin in connective tissues. So these are all examples of structural and support proteins that help cells and life to function and have a framework in them. So you can see how diverse the functions of proteins are in living organisms. Here is a quick video on contractile proteins.
Proteins, represented as purple blobs in this close-up of an animal cell, are the most complicated molecules known. A cell contains thousands of kinds of proteins, which carry out a variety of functions. In most cases, a protein's function depends on its complex three-dimensional structure. Let's see a demonstration of each of the protein molecules shown here and learn about its function. Observe how function depends on protein shape and changes in shape. Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins. Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals an animal's cells to take in sugar. The hormone receptor is also a protein. Signal molecules bind to receptor proteins that can then relay messages inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules are also proteins. Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell. Hemoglobin is the transport protein that carries oxygen in the blood. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response. An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells. Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos. Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal. Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings. The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction.
So a nice review of what we just talked about on the last slide. Hormonal proteins function in a variety of ways, and for the sake of time, you can go to the Mastering Biology study by chapter, and you can watch the hormonal proteins video if you have the desire to. So proteins are made up of 20 amino acids. These are our 20 amino acids. Some are nonpolar, hydrophobic. Remember, nonpolar means they are water-fearing. Polar, hydrophilic, mean water-loving. You have acidic and basic amino acids. And these amino acids are going to contribute to the tertiary shape of the protein once we get to that, and we'll look at the different shapes of proteins. So that is why it's important to note that you have nonpolar, polar, and charged amino acids, because they contribute to the way the final protein shape is. When amino acids are put together, this is how you make a protein. So amino acids are the monomeric units for proteins. And then putting many of those monomeric units together in a polymer is how you get a protein. And a lot of times you'll see a protein called a polypeptide chain, and that is because the bond that holds amino acids together is a peptide bond. Again, it is a dehydration, making a molecule of water during the formation of that peptide bond. So dehydration synthesis is how the reaction takes place to join the two amino acids by a peptide bond. There are several ways in visualizing proteins. You can look at space filling models, ribbon models, the wireframe model. All of these illustrate the folding of the protein. And as I mentioned before, that folding of the protein is determined by the amino acids contained within the protein. And most of our proteins are lock and key. Here's an example. Here we have an antibody protein that will bind to, and see how it fits with, a protein from a flu virus. So you made an antibody protein because you were either exposed to the flu virus or you were vaccinated for the flu virus. And the vaccination gave your body the ability to make this antibody protein to protect you from the flu virus if you ever encountered it. Here are our levels of protein structure, as I mentioned before. So our primary structure is just the single chain of our amino acids. So the primary is the unique sequence of amino acids held together by those peptide bonds. Our secondary structure consists of coils and folds in the polypeptide chain. These are called alpha helices or beta pleated sheets. And a lot of these are determined by hydrogen bonds. Our tertiary structure is determined by lots of factors. They can be determined by disulfide bridges, whether things are hydrophobic, hydrophilic with amino acids. The hydrophobic are going to point in. And our tertiary structure is actually our functional structure of a protein. So it is that tertiary structure that contributes and determines whether a protein is functional. And here you can see a lot of those interactions, the hydrogen bonding, the disulfide bridges, the R groups. Here's an ionic bond, so attraction between a slightly positive, slightly negative amino acid to each other. And then our hydrophobic amino acids are going to fold in to hide from the watery environment that they are in. So all of these different interactions contribute to the tertiary folding. And then tertiary proteins can come together to form quaternary structures, and that's interactions between multiple polypeptide chains that are in that tertiary structure.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of